Two co-chairs for the panel discussion session, Dr. Tian from AstraZeneca and Dr. Han from AIH to come to the stage as well. And please, do a self-intro to the audience. Thanks.
All right, so please come to the stage. Oh, the speaker. So sit here. Yeah. Yeah, it's fine.
3, 4.
4?
Yeah.
Yeah, let's see. There's another speaker. 最后我先收下，介绍到这个。Jenny Chen, yeah. This is right? That's fine. All right. Thank you, everyone. So I hope you enjoy this seminar so far. This is our last session. So it will take about 20 to 30 minutes. So if you do have a question, don't hesitate to ask. So we have, besides the 3 speakers in our session 2, we have other guest panelists will be introduced by Yixin.
Hello, everyone. Thank you for taking your weekend time to sit in our science symposium. It's a fantastic morning. And besides our 3 speakers, let's also introduce Dr. Jenny Cheng. Dr. Cheng is the Director of Regulatory Affairs at AstraZeneca. She is working on cardiovascular, renal, and metabolism projects as global regulatory lead and regional lead. Prior to her current role, she garnered the extensive experience in drug development and early clinical studies during her work at FDA CDER as a clinical pharmacologist for 6 years, and followed by 2 years at AstraZeneca. She also conducted research on GI liver disease models at the NCI-NIH and published over 20 articles in peer-reviewed journals prior to her industry experience. Let's welcome the panelists and provide your question, and we start the discussion. Okay.
I have a question for the Dr. Chen. You show your slides for that animal model testing, and that the male monkey, the results is not that great compared with the female monkey. Do you have any interpretation for that? And the second question is, how do you select these monkey models for your specific clinical purpose? And I think this is a very important. If you if you consider the further application, that you have the small animal testing already. And how do you consider in which case you need the bigger animal testing too? And sometimes you don't need the bigger animal. So all these cases different. Could you, from the regulatory viewpoint, to give some explanations? Thank you.
Thank you for the question. This is a very big subject, how to choose the animal, how to weigh the data, and the species, how to decide which species is the right one. Usually, if it's a standard testing, you need 2 species, non-rodent, 1 rodent, 1 non-rodent. So a standard package will have the rats, usually have the rats, just because of the historical database is very rich. So, and also because of the reproductive toxicity, the rats, the sprague-doli rats, they can produce lots of babies. I think 1 mother can produce up to 25 animals. So that is very good for the reproductive toxicity study for that reason. So people are using this for a while. So the rat has no big question. For the dog and for the monkey has asked the issue. If you can save the monkey, so the default is the dog currently, but you have to consider the relevance, the human relevance. So for the animal testing, it's all about the species relevance because the toxicity study basically uses the animal to predict the human. For biologics, the example I'm giving are polypeptides, amino acid. So they are biologics. So usually the specificity is very high for biologic compared with the small molecule. I mean, relatively. So for this case, there are two receptor get involved, GLP-1 and GIP. So the specificity need to be very high, very unique. From this perspective, the monkey is, they're all relevant, of course. The monkey is more appropriate for the animal study. But for the, for your first question, I only show one data for the monkey. It's not good or bad. It's body weight impact. I didn't show, I'm sorry. In the summary table, there's some monkey data, but it's not very significant. There are some, I think, a pancreas effect and body weight loss, food consumption is common for everyone. I think they put in the label. So pancreas is the target organ for both monkey and human. This is a good indication monkey is a relevant species in this case. And I think the label, I just read the label this morning. It's a very kind of, although not the black box warning, but it's a very serious indication in the label. Thank you.
Yes, I'd just like to compliment again all the speakers, but I have a question for Dr. Shao. Can you hear me?
Yeah.
Over here. Yeah, it was very nice, very nice.
Okay.
Very nice talk. I was wondering if you can comment on the triglyceride lowering effect of GLP-1 versus the triagonist. And the reason I ask is, as you probably know, that triglycerides have been disappointing as a target in terms of reduction in cardiovascular disease, but they certainly play a role in acute pancreatitis, and the FDA will approve a drug or use of drugs when triglycerides are above 500. And I know GLP-1 has a very modest effect on TG lowering. And so my question is, do the triagonists have a greater effect? And could that be a possible route of approval? Because I'm convinced that GLP-1 would be beneficial in the postprandial state. That's when usually trigger acute pancreatitis. But I think whatever excuse we can give for giving these GLP-1s or these triagonists, it would be wonderful. So question is, do triagonists do better than GLP-1 in triglyceride lowering?
In which indication? Sorry, I missed it.
Yeah. Do you know if the triagonists have a greater triglyceride lowering effect?
So you talk about the triglyceride reduction.
Yes, lowering.
Okay. Yes. I think in the animal studies, the triagonists may show some benefit for kidney disease, cardiovascular disease in animal study, but I don't think in the human that has not been confirmed yet. Right now, so far, it's still in the early stage.
Yeah, I can add one point. Recently we have several projects relevant to the TG correlation with the ASCVD, the cardiovascular event. So far, as you know, TG has not been approved as a key biomarker to show the cardiovascular event improvement, not like LDL-C, right? I know some efforts right now in the public try to put the TG together with ApoB and LDL-C together as a composite biomarkers to show the correlation. But for the TG itself, I haven't seen the strong evidence yet.
I just add a very small comment, and I showed like in my slides, like for the reduced trials resulted in the first FDA approved omega-3 medications. Well, that is for the total cardiovascular disease, like medications. And this time particularly point out the patients with high triglyceride. I totally agree. Now with the biomarker, that is a big debating like area, what should we put? For example, I also introduced the inflammation, like it's a high sensitive PCR, and they now urge to show it as a more independent as a predictor and risk factors. So I think now it's not on the available biomarker panels, but I still have things like in triglyceride and inflammation.
Yeah, if you significantly reduce the triglyceride, maybe double or triple, also avoid some clinical benefit like the pancreatitis, I think you may be reasonable, be a surrogate endpoint for approval. But for triglyceride itself, so far I think it has to be huge difference. So some genetic related to the triglyceride, very high triglyceride, be reduced can be approved now. Yeah.
The follow-up, I'm just curious, since GLP-1 has a central effect as well, is there any evidence that the use of GLP-1 changes the choice of food, high-fat food, high carbohydrate, high protein, and that, because when you try to manage these patients clinically who have genetic defects, it's very difficult. What we try to put them on is a 10% total fat diet, which is extremely hard to follow. And I wonder whether GLP-1 changes their perception of food and what kind of food is tolerable. Again, this is speculative, so maybe none of you have the answer, but it's a possibility, I think.
I think in the official label, the indication for either GLP-1 or semaglutide, you have to do diet control, regular exercise. If this behavior are not effective, then you take this drug to reduce your body weight. That's the indication. But actually right now in the real world, people just take the drug. They don't do some regular exercise. They don't do the diet control.
Yeah, thanks for the answer. So let's move to the next.
Yeah, so I have two questions for Dr. Yan. The first question is for omega-11. So you did clinical studies, and it looks like the results are pretty, good. So I'm just wondering, so do you have any like clinical outcome or event or endpoints type of observations? Because now, so it seems that the lipid profile got improved, but we don't know the real clinical outcome yet. So that's the first question. And the second question is, so you mentioned in your slide somehow omega-3 has been approved by FDA as a medication. And you also just said, but we know, so the dietary supplement, because FDA has a dietary supplement act that, because the regulatory pathway for those dietary supplement and drug is very different. Just wondering, so the omega-3 has been approved by FDA as a drug or medication, or is a dietary supplement. So it could be very different. So then, for those companies, if they try to do some studying, so those are the pathway is very different. Yeah, thank you.
Thank you. And those are two questions I like. So for your first question, since that is the first in human studies, and we want to first like for the safety and very simple, straightforward, and we want to see a significant outcome, but not a clinical outcome like some parameter outcome. So that's why we first set the goal as lipoprotein, which we achieved. And definitely what you said is 100% correct. I completely agree. Next, we need to recruit patients with some symptoms because the first study is healthy, right? Their lipid panel is normal and okay, you saw some like a trend. The next we want to recruit, for example, hypertriglyceridemia or hypercholesterolemia patient and see how those omega-11 really work. So that is a biomarker. And for the cardiovascular disease like event, that is much further advanced questions that need correlated with industries, how to formulate not as a grass, like generally considered safe, but also with a drug formula. And then you need to have a PK, PD, a series of study. That is a different, like overlapped, but that is a direction we should like consider. So now to the second question, that is a huge study in the field already for decades, medication and supplement about omega-3. So the significance of a reduced study, that is to answer the question straightforward, that is a drug, prescription drug. That is a purified EPA in ester, ethyl ester form, and with very high dosage, 4 grams. That is for the first, as you a moment ago said, biomarker is different from the events. That is to reduce events. Now, when we talk about a supplement, there's just too many brands available on the market and people get confused what to choose. As a matter of fact, very sadly, most of them first has very low dose of omega-3. When we talk about a medication, 4 grams versus you only get a 300 milligram, much less than 1 gram. And that is a combination, most supplement is a combination of EPA and DHA. And nowadays we know EPA and DHA overlapped mechanism, but also they have a separate function, especially in regard to cardiovascular disease. DHA actually, some studies showed increase LDL cholesterol, which is not that good. But how to optimize the ratio, that is another question. So your question, drug is different from nowadays supplement. And even when you take a supplement, you need to say, how's your baseline of omega-3? If very low, that may show some benefit, but you've already taken 2 times, 3 times a week, you eat fish, probably you may not just need it.
Yeah,
so they are for the details clarification. So next.
Yeah, thanks for the speakers. We need to announce that after this panel session, we will have a lunch session, and you will still have opportunity to talk to the speakers. And next, I received many requests for asking questions. The next question I will give to the audience who is asking Dr. Chen question. Who has a question to Dr. Chen? Okay. One question. Dr. Chen. Who has a question for Dr. Chen?
So if nobody has, I have one for Dr. Chen. So given the extended treatment time for diabetes or obesity, so how do you assess, or what is the best practice to assess the toxicity for this drug for used for cardiovascular disease or renal disease?
Okay, so so you're talking about safety, I think.
Yes, yeah.
Okay, let me think. You know, let's give an example. I think so far, you know, for obesity market, GLP is a very hot target, right? So, you know, it has been first approved for the type 2 diabetes and expanded to the obesity with a higher dose, and then expanded to the cardiovascular events, and followed by the renal, as illustrated by Dr. Xiao. So, you know, I think the dose selection is the key. And you can see like across all the approved drug, which has the you know, the CVOT and the CKD approval, they have the same dose for obesity indication, which means they have been used a higher dose for these high risk patient population. So I think they have long-term regarding the CBA study and CKD study, which is a longer study compared to obesity and type 2 diabetes. So they have a long-term safety monitoring, and they have the different safety monitoring plan. So I know right now almost every drug which will be approved for obesity or type 2, they try to extend their indication for the CVOT and CKD if possible, talking about GLP-1 mechanism action, because GLP-1 is relatively safe. And then it has been several approved drugs like semaglutide for these indications. So I know right now, like retatrutide from Lilly, Bema from Lilly, and even from AZ, from other Amgen, Maritide, they all try to move from obesity type 2 to the CVOT and CKD if possible, because FDA and the EU, they really want to see the potential benefit for comorbidity of these target patient population. And talking about from the regulatory to the market perspective, commercial guys in the company, they will have the win-win if you have these CVOT or CKD indication approved. So I think, it's efficacy and safety, but is really the trend for this market right now.
Thank you. For the interest of time, so we're gonna allow the last question. So please.
Yes, for Dr. Xiao, in his talk, I think you select patients, obesity patient by without diabetes. Yeah, why not to including diabetes? Yeah, also. So also you see a lot of effects of this new drugs for CBD or some related drugs. So I think in China there in the market now, that trend of the supplement industry. I think Tianshi Pharmaceutical did a clinical trial on Danshen, also for CBD. I think I heard that didn't get approved due to some experimental or design problems, or didn't reach a significant.
You ask Danshen or you ask
anti-obesity
drugs?
Yeah, your clinical design. So I think very important for
Danshen. The indication is for not for CV outcome. It's for the chronic angina. Angina indication. So they failed the trials because not failed the trial. They said 4 weeks is positive, 6 weeks has not been confirmed yet, which is different design from the FDA evaluated. That's for Danshen. Okay, back to your first question about the why they choose the obesity patient without diabetes. I think that's the marketing issue. Originally, I guess Novo Nordisk, right? The Novo Nordisk, they believe that patients with diabetes and obesity may not get a very positive result. So they originally choose the obesity without diabetes, but with cardiovascular problem. That maybe get a positive result. So they win the trial and they get the label. But in the second trial, in the kidney trial, they do the separate analysis, involve patients with diabetes, not diabetes, with the obesity and kidney, they with diabetes and the kidney problem. So in this trial, it's called diabetic nephropathy. So the diabetic complicated with kidney disease, and then they win the trial again. So I think that's the marketing issue originally, because semaglutide is the first drug approved with the cardiovascular kidney label. No more other drug get approved so far. So that's why originally to be very cautious. When you do the clinical trial, you want to get positive result. The straightforward, less complications may be easy to get a win. I think that's a step by step. Danshen is a different indication. Danshen is for the angina. And it's a short-term study. It's 4 weeks with 6 weeks study, because in the phase 2 trial, for the 4 weeks is not good enough. 6 weeks is good. So the phase 2 trial. So in the phase 3 trial, they design 6 weeks what come. So then they did not submit data to the FDA. They just make it public. They said we get 4 weeks positive, 6 weeks not positive. So it's the opposite it's different from the phase 2 trial. But FDA think you already defined 6 weeks is the endpoint time, and you win 4 weeks. You didn't win 6 weeks, so you cannot get approved. I think Danshen currently still ongoing, right? Do the cardiovascular study. It's not a CBA-USA trial. It's just for angina. It's improve the oxygen consumption. It's a little bit different.
For the sake of the time, let's move to the next step.
Let's welcome all the speakers and the panels for the great talks. And next, let's grant the certificate for all the panelists for their great talk.